thing. So I get the total details of the complete information of the cell containing the RNA, and that will be known as transcriptome. So every detailed information of the RNA, when I study the same, that will be transcriptome, and the complete molecular biology behind that will be transcriptomics. Similarly, when I take out the protein of a cell, if I go for the further detailing of the same, I will get the proteome, and the details will be proteomics. If the same thing, see, any kind of metabolite is given by what? The proteins which are present in the cell. And what is the metabolite? Okay, metabolites are those particular substances which are present within our cell, which actually go for the metabolic. Metabolites are actually the final form which we see within us, within the plants, within the animals, and studying them actually give us the details of all these things which were behind. So when we go the detailed version of the same for the different analysis of the metabolite, and whatever we receive at the end, that is your metabolome, and the study of the details of the same is known as metabolomics. And similarly, if you continue, you will get the complete phenotypic result of the particular organism. Now, this is what the complete omics will look like. Everything is interconnected. So when we start with DNA, we get the genome. When we start with RNA, it is the transcriptome, proteins, proteome, and the metabolites, that is the sugars, the nucleic acids, the amino acids, the lipids. Those are your metabolomes and the metabolomics, right? And finally, the total view of the complete organism, the phenotype of the organism, that is what you are visualizing at the end of the day, right? So everything, if you look behind, the metabolites are basically dependent upon what proteins are there. The metabolites are dependent upon what RNA is expressing. So everything is dependent upon the expression analysis, which is important for the final version of the phenotype that you actually see at the end of the day. And everything is interrelated, just as I said. So whatever you do, whatever studies you are going for, be it these studies related to the DNA sequence analysis, be it the proteomic related studies, be it the metabolic related studies, everything are actually interconnected to each other. And the essential part and center that you may see, that is bioinformatics, that is the computational tools. Considering the fact, the amount of data that these particular studies will generate, we cannot work upon them until and unless we have strong computational tools. So bioinformatics is a tool which will be linking all these particular studies, right? So what is the importance? Why we thought about this complete study, how they are important? Simple example, even I think a decade ago, when we started with the drug designing, actually it used to take so many steps to actually the FDA to approve a particular drug. And every scientist, they thought that probably it will take 2 to 3 years to develop a proper vaccine, because of the fact that even the human clinical trials will take almost 2 to 10 years. So how can we go for it? How can we find a proper drug for the same? So when we thought about the technologies which is actually helping out to shorten out the complete thing, you will see that every step is basically being concised because of the high throughput technologies which have come into being. So be it genomics, be it proteomics, be it the computational analysis, be it the high throughput screening, everything has taken a toll. And now the things which used to take almost 10 years, now they have shortened up within a small time period, right? So this is the reason why omics has got so much importance in our life, right? Now, being a botanist, I wanted to give an example of a plant. So this is Arabidopsis thaliana. So what exactly and how exactly we move ahead when we are planning to look into the supposedly this Arabidopsis thaliana has different colored flowers or different particular phenotype. So how do we go for it, that what actually is responsible behind its color or behind its structure or behind the kind that it is producing? The first job is to take out the metabolites, to take out the complete RNA of the same and to create the transcriptomes of the same. and that will be approximately around 20,000 genes. Now, after that, it is very essential that you integrate all these data. And when you are integrating all these data, basically you get means huge amount of data that needs to be concised up, right? So how do you predict a gene? You require computational analysis for the same. So once you get the transcriptomics, once you get the metabolomics, I will be telling you details in my lecture. You go for all these computational analysis, and once you get the gene prediction, आपको जीन पता चल गया कि हाँ ये जीन है, but then how do you confirm the same? You go for the proper wet labs for the same. You confirm them by the experiment. So if you see, they are actually experimenting into the fact that if it is a wild type plant, how much amount it is getting, and if it is a cultivar of this type, how much glucosinolate is being produced. So that means we are looking out for the metabolites which is actually being produced. And once we link both the data, we get the final answer that why the particular plant or why the particular organism is showing the phenotype the way it is being shown. So this is actually so much interlinked, and until and unless you are getting every data answers, you cannot decide that why the phenotype is looking the way it is looking like, right? So omics means a lot of data, and this is a complete revolution which took place in within 2-3 decades. So we started with genome, then we moved ahead to transcriptome, then proteome, then metabolome. So we had every details and every data in front of us to finally decide the fact that how the omics are so essential for us, why omics revolution is so crucial for our existence. So coming to the details of the same, let's start with genomics. So what is subdiscipline of genetics, which is devoted to mapping, sequencing, and functional analysis of genomics. Basically, we try to understand all the genes which is present in an organism, right? So it will involve the study of all the genes at the DNA level, at the mRNA level, at different levels, so that there can be a detailed version of what this cell will carry. So whatever cell we are receiving for the detailed study of in the genomics, we will get the details by studying in these particular variations, right? So if I talk about the difference between genetics and genomics, genetics is very different considering that it is the study of hereditary, whereas genomics is what? It is the study of entirety of the organism's genes. Genetics refers to a specific sequence of DNA on a single chromosome, whereas genomics is complete genetic makeup of an organism. Genetics will be involving the study of function and composition of a single gene, whereas genomics, it will be all the genes that we are going to address, as well as their interrelationship amongst themselves. So that is the reason why genomics is so different from genetics, right? So when we talk about the types of genomics, we have a few types. Let's start from the most basic one, that is the structural genomics. Please try and understand. I have a lot to cover, so at times I might be going a bit fast, thinking that you are already knowing. But if at all you think that I am too fast, just unmute and let me know. Okay. So if I talk about structural genomics, basically I'm trying to know the structure of the chromosome. So what exactly is the position of the gene? How is the chromosome looking like? So we go for the different kinds of mapping for the same. We even go for the whole genome sequencing, which can be possible by so many different techniques. I'm not talking about gene, I'm talking about whole genome sequencing. Also, we can go for the map position based cloning of gene, that is chromosome walking, chromosome jumping. So basically, we are trying to understand the structure of the genome, right? So structural genomics is what? It is the organization and sequence of genetic information which is contained within a genome. So early step in characterizing a genome requires a prepared genetic and physical map of its chromosomes. And these maps will actually give you the detailed information about the position of the genes, the molecular markers present in the chromosome, the chromosomal segments which are actually essential for positioning them and aligning the stretch so that we go further for whole genome sequence. Essentially speaking, not it is right now that when I can say very confidently that most of the labs, means I'm not very sure, but most of the labs of molecular biology are carrying their own sequencer. So whenever we strike upon a new thing, we just directly go for the whole genome sequence, and overnight we can publish a paper. But that was not true 10 to 15 years ago. We used to work at the gene level, and before that we used to work at the chromosome level. So try and understand that how we have advanced with each and every step. So earlier we used to go for these mappings to know the structural details of a particular genome. So it was very essential that how you map a genome on the chromosome. And this was possible by physical mapping. So we used to look out for the overlapping fragments. We used to position it, and we used to tell that probably this area is containing the genetic marker. Probably this is the structure of the chromosome. And how we used to move ahead? We used to finally decide upon the fact that we have to go for the whole genome sequencing. That is the most advanced version. So what was the basic tools with which we started? Earlier, since we didn't have a lot of tools, we used to go for clone by clone method. And now whole genome shotgun method. And now, of course, if you get to know better, then next gen sequencing. So every step had certain drawbacks because of which we moved ahead, getting better results, getting better answers. So if I talk about only whole genome shotgun technique, it was announced by Venter in the year of 1998, where he had actually got into a company of cellular genomics in which he privately tried to sequence the human genome. See, when the Human Genome Consortium was set up, there were many people who were working independently as well, because it's a fight. Whosoever will publish it earlier, they will get the benefit of the same. So every single person involved in the molecular biology of the Human Genome Project, they can understand what I'm trying to say. Basically, Venter had its own company. So he tried his efforts so that a human genome can be sequenced. And he proposed this new technique, which was the shotgun sequencing approach. What he said was that it is a technique which will be involving randomly breaking up DNA sequences into lots of small pieces and then reassembling them by looking for regions of overlap. Now, I will explain it to you with a small example. See, this is the genome. जब हम DNA extract करते हैं, so this is the coiled version of the same, that is the genome of that particular cell. So what essentially is done in the whole genome shotgun method is that they cut it by certain REs, restriction enzymes, so that we get many small overlapping regions. Overlapping means kahin na kahin unka overlaps ho, right? So it's not a complete stretch. They require certain overlaps. Now each fragment is then cloned in bacteria. Why do we go for cloning purpose? Why not PCR? I will be telling you. So whenever there is cloning, what we do is that we get multiple copies of the same thing. So when we are trying to clone this particular fragment, we are getting multiple copies of the same. And once we are getting each and every fragment multiplied in several folds, it's easier for us to sequence the same. Once sequenced, the final job is to get the entire genomic sequence. And how do they do that? By using computational analysis. They're powerful computer programs. What they do is that they will find the overlaps and they will say, yes, this is the segment A, this is the segment B. Why? Because a portion of A and B are overlapping. Secondly, then this is C, then this is D, and this is how we get the complete stretch. Now, this is what whole genome shotgun sequencing means. This is the write-up of the same. You can go through it. I just tried to explain the same with the diagram. And advantages, huge number of advantages. As I said, this is better than the clone by clone method. Reason behind, because this is a faster process. This is actually better in the sense that we don't require a lot of DNA for the same. A small amount can give us the complete genome sequence. And in particular, this is very, very efficient. So whenever we are assembling and we are getting the final genome, we actually do not require a lot of data pre-hand before that, right? So this is considered to be a very faster and less expensive method than actually going for clone by clone method. And if I look into the clone by clone method, it is having a bit difficulty, but then this was started earlier. This is the reason why this will be having certain, drawbacks of the same. So what they did was that whenever we are going for clone by clone method, we start firstly to actually find the entire chromosome. We should have an essential idea of the entire chromosome before going for the sequencing. So what they do is that they will create large insert clones so that if they are assembled in a proper way, they will get the entire chromosome in order, right? So first thing, they will create large insert clones. Second thing, these large insert clones are then again broken up so that they can be created into small, again, overlapping fragments. And these overlapping fragments are further utilized for the purpose of, multiplication. Over here, we require bags for the same. I hope you know what bags are, bacterial artificial chromosomes. So we go for the same, we multiply the same, and then we go for the sequencing. And once we get the sequence, we require enough overlapping these small fragments so that they can be, overlapped, and we get the final version of the whole genome. And how do we do that? We actually put together the first step and the last step of the overlaps. So until and unless we get the first map, which is already existing, after that we fill up those things. At times we are left with certain gaps, but then every technique has its own disadvantage. We cannot discuss about the same in this particular session. So what exactly is clone by clone? We get a genomic DNA, we fragment it, we get the large insert clones, and then we are inserting these DNA fragments in the back. Again, we are fragmenting the small areas. These are small insert clones. We are cloning them and finally sequencing them. This is what a sequenced data looks like. And once we see, these are in millions, so depending upon the organism. And once we get them, we actually find the overlaps and we create the whole genome. So this portion is totally computational analysis. This portion is completely wet lab analysis. So these are the basic ideologies by which we actually create the whole genome. Means earlier when we used to talk about एक gene sequence कर लिया बहुत बड़ी बात है, so let's submit it in the NCBI, big thing, big job. Now the complete genome is getting sequenced. Oh, it's a normal thing, it hardly takes a day or two, please submit it. So the situation has changed so well because of the advanced technology that we are working with. So now coming to the functional genomics, and another part of genomics, as the name suggests, functional. As I said, it's not necessary that if you know what is the ATGC of a particular gene, it's not necessary that you end over here. How does that matter that if you get the whole genome, how will you know that what this gene works for? आपको नहीं पता कि ये gene किस काम का है, until and unless you go for the functional genomics. That means you are trying to know the function of the gene. And how do you go for that? You go for the transcriptomics, you go for the proteomics. Basically, you try to understand that what actually the gene will look out for, what actually the gene will function for, right? So why do we need to study it? Basically, when we sequenced the whole genome, there were many open reading frames, ORFs, basically, which were not giving any answer that what they were coding for. There were almost 30% of ORFs which didn't have any kind of function reported. So people were quite confused that if they are present in the genome, then they must be going for some kind of function. And what are those functions? So we need to study the functional genomics to know the answer of the same. For example, there can be a single gene which can give rise to multiple gene products. Means a single gene can code for many particular phenotypes. So where exactly the situation is changing? They are changing at several levels beyond. So it can be possible that the RNA is alternatively spliced. It can be possible that during the editing of mRNA, there is situational changes because of which the changes that you are visualizing in the phenotype, that is very different. It is quite possible that at the post-translational modification, they are creating a lot of changes. So where exactly the gene that we have talked about in the whole genome, where exactly they are moving for the, finding its function, that is very essential. And that is why functional genomics, where it is expressing what exactly is the phenotype, that is essential to know the final version of the genome. So that is the reason from gene to the function of the gene, this complete study needs to be done. Until and unless you do that, only the study of genome won't give you all the answer. Now, there is a third type. In fact, there are thousands of types. I cannot cover all, but let's go ahead with few of them more. So there is mutational genomics, in which you actually study for the mutations. You go for that part of the genomics where certain genes will be associated with certain mutations. And this will be based upon the fact that what genomic, what epigenomic, or what transcript alteration is happening within the cell. And it is quite possible that because of certain changes during the processing of the particular gene, you have got the different kind of mutation. And this is the reason there is a necessity for a mutational genomics. There are three ways by which they go for. First way, where you are mutating every single gene present in the genome. So if you have a genome, you are mutating all the genes, and then you are generating the banks of that specific mutant strains. One by one, you go for the systematic approach. Secondly, you go for a random approach. You mutate it not knowingly that what actually you are mutating. So then you go for the phenotypic study, then you match the same with the sequence database, and then you get to know that probably this gene is attached with this function. So mutating it beforehand randomly also gives you the detailed data. Third approach, in which there are many techniques involved, in which you generate functional phenocopies of mutant alleles. There is a likeliness of a mutation without actually altering the DNA sequence of an organism. So as we already know that mutation not necessarily can be inherited, can be creating a phenotypic difference. So it is a requirement that what mutation actually you are going for. And you already know that there are certain studies like oncogenomics where mutation actually plays a very, very big role, because whenever you have an accumulation of DNA mutation, it can lead to cancer. And the study of the same cancer associated genes lead to oncogenomics. So the goal of oncogenomics is to identify new oncogenes or tumor suppressor genes that might provide into the details of what actually the cancer is being caused of. So it is very essential. We already know that it's not feasible on our part to cure cancer at the final stage. So people are moving towards molecular biology to actually stop it at the level higher. So it's important that we diagnose this cancer beforehand. We have started using molecular markers for the same. If we get to know that there is an cancer, we just try to look out for whether a marker is present or not. If a marker is present, that is the early detection of cancer. We can also go for classifying the cancer depending upon those markers. And of course, if we are knowing the fact that there is a particular mutation present within the particular organism, they can always go for the treatment beforehand the multiplication starts. So all these are the reasons behind the goal of oncogenesis to be essential for us to go ahead with the studies. Now there is one final version, that is the comparative genomics. This you must have heard of. Here we are trying to compare the genomics of different organisms. It's very important to know how one organism has evolved from other. And until and unless we compare the same, we cannot know the evolutionary conserved sequences between two different organisms. Of course, we go for computational analysis over here as well. And how this is important? Well, they go for understanding the fact of how the evolution has happened, what genes are important, and which genes are basically conserved among so many species or similar type of species, which gene is unique to a particular organism. All these things is feasible when you are comparing genomics of different kind. So this is feasible because you get to know the comparison between different organisms, their genome size, the number of genes, the chromosome number which is present, and everything like that, so that when you are aligning the particular DNA from different species, you get to know that how the genomes are different from each other, right? The tools, as I said, they are the computational tools. That is, nowadays they are known as bioinformatics. There are several other tools by which we can actually analyze the data, and we can actually tell that how the homologs are different from each other, where exactly the conserved sequences are situated. So these are the applications of comparative genomics, where they are looking out for an approach to know that how several related pathogens can be, leading to the development of vaccines. How they are important, that if we know the details of any particular loci, how they can be essential for breeding crops. So applications of comparative genomics is very, very, important in today's world. So coming to the next field, that is transcriptomics. As I said, transcriptomics, actually we actually read into the transcriptome of an organism. And why transcriptome? Starting with genomics, where we have talked about the complete set of DNA of an organism. So why transcriptome is so essential for us? See, whenever we talk about expression analysis, we start from the level of mRNA. We start from that level because of the fact that not necessarily all the genes which are present in a genome will be expressing in the final form. So transcriptome is what? It is the complete set of transcript in a cell and their quantity for a specific developmental stage or physiological condition in a particular time. So that means it is limited by time. So it's very essential to understand your transcriptome. A plant's transcriptome can vary when you are collecting the sample. If you are collecting the sample early in the morning, and if you are collecting the sample late in the night, you will see there is a variation of RNA. So why this is happening, what actually is the role of transcriptome, that I will be discussing now. So basically transcriptome is very essential when you are trying to understand what is the functioning of the genome, how the molecular constituents of cells and tissues are important, why and where exactly transcriptomes can be utilized for disease and development. So transcriptome is what? It is the study of RNA in any of its form. So it can be mRNA, rRNA, tRNA, it can be any of the RNA, it can even be fragment of RNA which might have broken off. So it can be any form of RNA which you have extracted from a cell at a particular time. That will be the transcriptome of that particular organism, right? So transcriptomics is what? It is the study of transcriptome. Okay. So why is it essential? It is very, very essential considering the fact that when you have a particular gene, you know that there will be a regulatory sequence before that, there will be a ORF region, there will be a regulatory sequence behind that, and that is the complete gene. But when you are going for transcription, when you are creating RNA from that, you will see that it has shortened up in size. So what has it thrown behind? It has thrown behind many of the factors which are not important now when we are moving ahead. So when we move ahead, when we see the mature mRNA, you might see that they are only carrying those regions, that is the exonic regions, which will be coding for a particular protein, right? So the leftover area is the protein coding region. And when you see the mature mRNA, you will see that there is a capping being done, there is a tailing being done, and we have got a concised RNA compared to the genome that we started with, right? So that means that RNA that we started with, that we are working with, that has been modified at various levels. And this is the reason why gene expression is so important, right? So what is gene expression? When we are studying whether the gene that has been expressed finally into the phenotype that you are looking into is exactly the RNA or the protein that we are finally working with. So at various levels, this gene expression can be controlled. You already know of these things, but basically when we are talking before transcription, it can be epigenetics, it can be transcription factors which are involved. Beyond transcription, once there is splicing, you will get a different kind of RNA. Once there is capping, once there is tailing, if there is any kind of change, you will get post-transcription changes. Then there is silencing. It is quite possible if the microRNA is binding to the transcript, it is inhibiting the complete translation process. So it is quite possible that post-transcription, there can be huge number of changes, because of which when you started with the genome, which was huge in number or huge in size, it is quite different from what you are seeing the phenotype as being expressed by the RNA and the protein. So post-translation, it can be signal peptides, which is actually the reason why the protein is being shipped So where exactly it has been cleaved, what exactly the modification has happened during the signaling process. There can be post-translational modification so that there are covalent changes that the protein is altering the function. That means that when you have started with all the genes of the genome and when you are finally visualizing the organism, there is a huge change in the expression. And please remember, the expression analysis has taken a toll on the fact that when we are working, it's not necessary that what we are reporting at gene level is true at the protein level, right? So there are complex controls which are happening within the organism. And this is the reason why I keep on asking my student that even though from your body, if every cell is carrying the same genome, do you think that the liver cell and the nerve cell will be having the same transcriptome? And the answer is no. They are having the same genome. Yes, very much true. But since they are acting as a liver cell and since they are acting as a nerve cell or a muscle cell, they are having different transcriptome, different proteome. And at what level they are different? Well, there is complex control which is going on throughout the process, which is actually causing the differences. Right? So that is why transcriptome study is so essential. Now, why would transcription vary? It can vary from series of differences. It can vary because of the drug you are taking. If you are having less sleep, if you are having more sleep, the transcription can vary. Developmental stages, a child will have a different kind of transcription. Myself will have a different kind of transcription. My father will have a different kind of transcription, right? So developmental stages can change the requirement. The kind of energy my child will require, I won't require, and my father will be having the least amount of energy. So where exactly you can find the difference? You can find the difference at the transcriptome level. Tissues need different proteins for functions, as I said. You can extract the tissues at different levels. You will see that the genome is same, but the transcriptome is different. If you are having diseases, the diseases actually distort the healthy processes. The normal healthy human being, if you carry out the transcriptomics for the same, and a diseased human being, if you are carrying the transcriptomics of the same of the diseased human being, you will see a varied difference between the two. Even though at genome level, if you genome map both the organism, you might get absolutely 99% identity, right? So we need to measure the mRNA copies in a sample so that we know that what is the possible transcript. Now, this is one thing which is very, very essential. That is quantification of the mRNA copy. In the sense, as I said, my child might be having one mRNA which might be produced at a very high level. I am carrying the same mRNA which might be produced at a mediocre level, and my father is carrying the same mRNA which is being produced in a very low amount. So measuring the amount of copies of the mRNA which is denoting for a particular phenotype, that will tell you that why exactly the phenotypes are differing. We all are human beings. We if you look even into the same species plant, we will find that the transcriptomes are varying, right? So this is the reason why transcription varies, and this is the reason why expression analysis is so important. So what are the takeaway messages? First, the genome found throughout an organism is a constant. That means every cell will have a specific gene which will be true throughout the organism. But when we go for gene expression, which is known as transcript, we need to go out for different kinds of tissues, and we need to go out for different technologies to find out the differences. How we go for that? Microarray, which is a hybridization technique. RNA sequencing. This is a very advanced technique in which we directly sequence the RNA. So basically, these are the most essential forms of technologies which has helped us into know the details of transcriptomics. So what are the aims of transcriptomics? We need to know the details about mRNA. We need to know what are the non-coding RNAs, what are the small RNAs. Also, we need to determine the transcriptional structure of the genes in terms of that where it is starting. What is the 5' and 3' end? Where exactly the splicing is happening? What post-transcriptional modification might have happened? So all these things are essential to know about in the studies of transcriptomics. And finally, to quantify, please remember to quantify the changing expression levels. Each transcript varies during the development and under different conditions. So whenever we are trying to know the changes of the expression level, we need to quantify the amount of RNA produced, and because of which we have wonderful technologies in front of us, right? Now, there is one more thing I thought of sharing with you, because mRNA, whenever we are studying transcriptomics, it's not so stable. This is the reason why you will observe that each of the technologies that we will talk about, they are requiring cDNA instead. What is cDNA? Can anyone answer? What is cDNA? Are you guys there? Okay, let it be. So whenever we are talking about mRNA, if I go for reverse transcription, we can actually produce DNA in a single strand form, and that DNA is very, very essential. Why? Because it is very different from the normal DNA that we work with, right? So it won't be having all the intronic regions. It will have only the coding regions. But since they are not degraded by the RNAs, which is present every single place, that is the reason why cDNA is very, very essential for all the transcriptomic studies. So when we go for high throughput transcriptomic profiling, we actually go for looking out for the mRNA, which will be getting us the transcript idea of the cells. We will also be moving ahead with the microarrays, which will actually help us into knowing that which cDNA can be is actually present when we see. I will discuss microarray because this is very essential. Microarray is a technique in transcriptomics in which we actually look out for a particular cDNA which has been fluorescently labeled. And when we take out samples from a single human being, we try to find out which one has actually fluorescently given up. And when we measure the amount of fluorescence it has given out, we can even quantify the same. And of course, the sequencing technique, which is the most advanced techniques for transcriptomics, that we can directly determine the cDNA sequence. So by reading the count of the same, we can also know that how much amount of RNA is present. So what is microarray? I wanted to tell you exactly. This is what the microarray looks like post fluorescence. So supposedly we have a 96 well plate. Each spot will be having a different DNA probe, and they will be, the microarray plate will be consisting of DNA probes which will be fixed to a solid support such as a nylon membrane or a glass slide. So what exactly are they? They are the DNA or the cDNA we have extracted and we are essentially interested in. So whenever we are extracting the RNA from the cell, we are labeling them with a particular fluorescence tag, and then we allow them to actually complement with the probe. Wherever it will find is complementary, it will flourish. So you can see the final version of the same. This is a 96 well plate. At sometimes you will see that there is a green color fluorescence, there is an orange color fluorescence. They are depicting the fact that whether that particular DNA is having that particular probe or not, whether it is matching or not. And it is so simple. If it is fluorescing more, that means that it is having more amount of transcript. It is fluorescing less, it will be having less amount of transcript. Means RNA ज़्यादा produce हो रहा है या फिर कम produce हो रहा है, ये उसके amount of coloration से पता चल जाता है. So this is one technique which is utilized these days so much for actually identifying the fact that whether a person is diseased or not. एक छोटे से disease के लिए भी हम लोग human genome को बस extract करते हैं, tissue sample लेते हैं, blood extract करते हैं, and we just directly go for the microarray analysis, and we can find that whether a particular disease which we are actually using as a probe, whether that is fluorescing or not. And if it is fluorescing, we declare that that person is diseased or carrying that, or positive, or that person is carrying that particular gene of disease, right? So this is the normal DNA microarray experiment. I need not go into details of the same. I've just told you that we directly isolate the mRNA, we go for cDNA, and then we label these cDNA by fluorescent labeled nucleotides. We hybridize these labeled cDNA to DNA microarray, and then we wash and scan. And we observe the fluorescence, how they are fluorescing. Coming to the fact that how transcriptomics are so essential. First thing, they're important in creating ESTs. What are ESTs? Express sequence tag. See, when we are talking about complete genome, not essentially this will express. ESTs are what? They are small stretches of genome or gene, which will surely express in the final version. And this is very, very essential for plant species, for any organism. And reporting EST, that means we are reporting the amount of particular RNA, whether it is expressing or not. And this is so essential. Why? Because when you create primers out of them, you want to look up for a particular gene, you want to go for the breeding purpose, you just take out the EST, you design primers, and you move ahead. So EST's finding is so essential, and this is possible by transcriptome assembly and profiling. Secondly, for finding out about the small RNA characterization, something very commonly found and known, I think 99% people might be knowing what gene silencing is. And you also know the fact that how it is essential for creating a particular variety where you want to silence a particular protein or a particular gene. So what you go for microRNAs, you go for siRNAs, and they are important for creating the regulatory role in actually stopping a transcript or stopping a particular gene, and that will lead to silencing of the gene. And this is so essential, and this can be possible if and only if you go for detailed RNA studies, right? So these are the small RNA characterization, and of course eQTL, you must be knowing about QTL mapping. eQTL is what? It is expression QTL. So any particular QTL which will surely express, any particular QTL which will give into the phenotypic data, that means it is important for us, right? So any particular QTL which will be important for the metabolite, which will be important for any of the processes. So we require at that level the data. And for plant breeding, it is so essential to get eQTL, right? So we actually go for the level of association mapping. We associate the QTLs with the phenotype that we are observing, and we can create a new cultivar with the same. So all these detailing of the transcriptomes are so essential for knowing the details of a particular gene, right? Coming to the advanced version, proteomics, as I said, this is something very related, but again, very different, because now we are moving away from nucleotides. Proteomics, as I said, it is study of proteomes. Now, proteomics terminology was declared very late. It was by a postdoc fellow, Mark Wilson, and it was essential. Why? Because proteome was considered to be very essential to know the details of any cell's expression. So whenever at a particular time a protein has been expressed, the total set of protein expression needs to be studied, and this complete detailed study of the same is known as proteome. And the molecular biology behind it and the study and the techniques behind it led to the development of proteomics. So basically, be it genomics, be it transcriptomics, until and unless you levelize the whole thing with proteomics, you are at a loss. So why you are at a loss, I will tell you in details. Because of the fact that not necessarily or not essentially a protein can be expressed even if it is present in a gene, right? So it is very essential that if a protein has been expressed, we need to know that how much protein has been expressed, what actually techniques can be utilized to study the protein products of the genome. And of course, one important part, proteins interact amongst themselves. So until and unless we go into the detailed study about their interactions, we cannot know the details about proteomics. So there are basically two different methods which we'll be discussing. One is two-dimensional electrophoresis, which actually allows us to separate the complex protein mixtures based upon mass by charge, or mass and the molecular mass and the isoelectric points of the proteins. And there is mass spectrometry, where we can identify as well as we can go for the structural analysis of the particular protein. So when we started, we knew that one gene was very important. We also knew that one gene gives one protein, but it is no more true. It is no more true. We now know that one genome can lead to many proteomes. We also know that one gene is no longer equal to one protein. So how many proteins can they be equivalent to? We don't know. So basically, when we talk about the chemical diversity, the genomics have only four bases by which we can start with ATGC, right? When we move ahead to proteomics, it gives us differences in the different kinds of proteins based only on 20 amino acids, and metabolomics, it will give you more detailed version. I will go over there as well. So that means from genomics, when we move ahead to proteomics, the chemical diversity is increasing. So where there is one genome of an organism, it can be essentially invariant in all its cells. That means all the cells can have a different kind of proteome. And this is so true as far as the axis is time. As I said, time is very essential. So sorry. So when you talk about time, you know that the proteome can vary, you know that the transcriptome can vary. That means the complete expression can vary, right? So whenever we talk of any protein product of a single gene, it is quite possible that it can be in multiple forms because of so many modifications that I just discussed with you. The modifications can occur at the transcription level, it can occur at the translation level, it can be because of mutation, it can be because of so many reasons, that when you are talking about one gene, it's quite possible that you can get many proteins. And we don't know how many proteins. We really don't know until and unless we go for the proteomics. So genomics, where they cannot answer all the questions of yours, all the queries of yours, I am very much sure when you go at the level of proteomics, you will get at least 99% of your answers. So why is it so? As I said, there are so many regulations that are happening in between. You see, there are so many regulations. And when you are going for all these regulations, the final version of protein, when you go for that, it will bridge the gap between the genome sequence and the final phenotype or the behavior of the cell that you're observing at present. So whether the difference is because of this particular control or this particular control or this particular control, that you won't get answer of, but surely you will get an answer by the phenotype observation that what exactly it is expressing. So this behavior difference between the gene products, it is very, very essential to study proteomics for the same. And this is because of the fact of so many cellular differences that happen post the process of replication that it requires to be addressed, right? So what exactly proteomics helps in? They help in the identification of the protein. They also help in the expression studies, functioning of the protein, knowing the post-translational modification. They help us in knowing the localization and compartmentalization. They tell us the protein-protein interaction. In fact, every answer of genomics which we are left behind can be answered from the proteomics, right? So how do we start with? We start with the expression comparison. That means we start with the protein of two different samples, how they are different from each other. Secondly, we go for the simulation of the same. We map out the three-dimensional structure of the protein and we try to find out that where exactly the change has happened. And finally, we try to know that how the proteins are interacting. What are the three-dimensional structures of the protein? How the cellular localization has happened post the translation. So every post-translational modification or the PTMs that has happened for creation of the protein, those answers are received after the studies of proteomics, right? So what exactly we do in proteome analysis? We take out the information in the sample. We try to study at the expression level. We try to know out the three-dimensional structure of the protein, and we try to understand the interaction between proteins and proteins. And you must see that everywhere there are computational analysis involved. So even if we are requiring the data in the form of wet lab, we require the dry lab somewhere to actually connect all the dots to find out the final proteomic analysis. So what is the proteome analysis process? We get the protein mixture, we can digest it, we get the peptide mixture. We separate it and we will receive the peptides in form of separated form, either on the based upon mass or based upon the charge. and then we go for mass spectroanalysis. The data that we receive, they can be utilized by they can be reduced by the algorithms, and they can be identified. Or else we can directly go and separate the proteins, and then we can go for digestion and then follow the same process, right? So this is the general scheme for proteomic analysis. Why it is essential? As I said, at every level, whenever you are talking about a functional protein or the final biological system, it is essential that you know that what exactly went through, means what exactly happened to the genes while they were jumping from one stage to another, so that we know that what the phenotype looks like, right? So at every level, the analysis are essential. So what we start with is that we try to take the samples, and we create the two-dimensional electrophoresis, so that we get an image of the same type, in which we actually separate the proteins depending upon the mass and the charge. Once we run it for molecular mass, once we run it for the isoelectric point, and finally the dots will decipher the exact location of the particular protein, how much it has run. And then we go for the mass spectrometry after the gel excision and digestion. So mass spectrometry actually gives us the detail about what protein it is, right? So amongst the mixture of the protein, we can identify the exact protein. So I will be going into details of the same. So the first step or the first process is extraction. When we go for the extraction of the same, we go for the quantification by using two-dimensional gel electrophoresis. With digestives, we go for mass spectrometry, or else we go for the other process as I have discussed. Now, this is the two-dimensional gel electrophoresis, in which you can see that each dot is actually a different protein, and they have run based on two different factors. Once they have run based upon their molecular mass, second time they have been run based upon their isoelectric point. So every protein, based upon their exact location, if we know, or if we have a control of the same, if we know that if the position is this, that means that this protein is present. If the position is this, that means that this particular protein is present. So by actually comparing with the control, we get to know that which exact protein is present. And it is very, very important to get the exact protein from the mixture of the proteins that we are having. So once we run it, we can easily digest the gel. We can actually cut and get the sample by digesting the gel, and we can go for mass spectrometry. And in mass spectrometry, by actually running the complete process, or by running the complete analysis, depending upon the fact that how they are bending, I don't know how many of you know the mass spectrometry, depending upon the fact that how much they are bending upon the mass by charge ratio, we get peaks like this. And they are compared again with the control telling us that how which protein is present. Again, the peaks uh, longitudinally, how much they have run, they also tell us the quantification of the protein, that how much amount it is present. So it is very, very helpful to us to actually get the data from mass spectrometry to reduce the complete. See, when we started with a mixture of peptides, and when we are ending with, we are actually getting single proteins, and we can easily decipher that which protein is present and how much it is present by the mass spectroanalysis. So with this, we whatever data we are getting, we finally go for the computational analysis. Why? Because whenever we are talking about a cell, they are having hundreds and thousands and millions of proteins. It is very important that how they are interacting with each other. So if one protein is here, one protein is here, we try to create the interaction maps. That is, we create a grid of the same, so that we understand that what is the actual interaction amongst protein A with protein B with protein C. So how they are interacting with each other within the same cell, right? So once we get that data, our goals are achieved. And what are those goals? It is very simple, that it will help us in knowing that what actually has been expressed in your body, what actually has led into the detection of the connection between broad cellular function and pathways, right? And until and unless we get the details of proteomics, we cannot go into the further studies. So when we started with proteomics, there were certain challenges that we met with. That is that certain proteins are very bulky in nature. They can have more than 1,000 amounts of variants. It is quite possible that one particular gene is going for many types of protein. So it was not possible for us at that time because the analytical tools were very limited. And because of the normal general technique for absolute quantification of protein, we actually lagged behind it into certain data of expression analysis, right? So if I summarize the proteomics in details, basically we start with a sample and we actually prep the sample, be it a fluid or be it a blood sample, and we extract the proteins of the same. We go for two-dimensional page, and then when we are digesting, supposedly I'm interested in this protein, I take the portion of that gel and I digest it, I get the peptides, I go for the mass spectrometry. And then I can essentially interact with other techniques. It depends upon me that how much protein is there, which exact protein is there. So I can always go for other techniques as well. And finally, I can go for computational analysis to find the protein-protein interaction in the same organism, right? So this organism, even though their genome was this huge, not necessarily that everything will be found in the two-dimensional page. So proteomics had its own importance, and this is the reason why the proteomics study is so important, right? Now, until and unless I complete the metabolomics, my session won't end. So this is the last portion of today's lecture, that is metabolomics, as I said, study of metabolomes. Now, metabolomes, I also said that why it is essential, because of the fact that what genome is expressing, what RNA is expressing, what protein is expressing, not necessarily you get everything in form of metabolites. And until and unless you get it in the form of metabolites, you cannot understand the phenotype that you can see or you can visualize as a layman, right? So what is metabolomics? See, every definition and everything changed from the point that we started with. We started with DNA. We said that we are talking about RNA. Why? Because DNA alone cannot justify or cannot tell you the exact potential of a cell. So what exactly is essential? RNA. We also said that even if RNA is there, there is quite possibility that at the translational level or post-translational level, so many changes have happened that at the protein level, you get a different kind of protein compared to the DNA. So that means proteomics was important. And now I suggest that if I talk about metabolites, until and unless I finally discuss about what metabolites are being produced by these proteins, nothing is complete, right? So what exactly is happening within your cell? What exactly is happening within your body? That is dependent upon the factor that which particular metabolic pathways are happening within your body, right? So that means that metabolites which are involved in these pathways, that are important. And from where we are getting these metabolites? From the proteins that are being expressed, right? So when we started with genomics, why metabolomics was so essential? When we started with genomics, it was only 40,000 genes that we were talking about. We moved ahead to transcriptomics, and I said that it can be huge number of transcripts and can be possible from the same genome. So almost 1,50,000 transcripts, and proteomics more than that, 10 lakhs proteins. But when I talk about metabolites, not necessarily all the metabolites might be functioning in your body. So a lesser number has been produced. It is quite possible. And the study of this particular metabolites is known as metabolomics. And that is the reason why, until and unless these have been studied, you cannot understand the phenotype that you are seeing on a daily basis, right? So you know the fact that environment plays a huge role whenever we are talking about any plant, whenever we are talking about any human body or plant body or animal body. So if there is a phenotypic trait which is showing so many variants, it is not quite possible that you get the answer at the DNA level. It's quite possible that you don't get the answer at even the gene expression level. So at the level of biochemical pathways, you get your answers, because all the genes that have been expressed in the form of proteins, what kind of metabolites finally they are creating answers the phenotypes that you are receiving at the end of the day, right? And studying of metabolomics actually helps or sorts out all the problems that you meet with in the all the rest of the studies that I just discussed. So there is a integration which is possible by the simulation which happens with the pathways because of the metabolites that are present in a cell. So the cell having all the metabolites at a given particular time, again I say, I repeat myself, at a particular time will actually tell you the metabolome of that particular organism. Just a second. Okay, so and the study and the techniques involved in the study of metabolomes will be giving you the details of metabolomics, right? So any analysis, if you see from the beginning, I kept on saying all of them are interlinked. And until and unless you go into the details of these interlinks, you do not get the final answer, right? So if I look into the environmental influence, the environment influences the metabolites at the maximum level. So you may see that at the level of genomics, the environment doesn't influence that much except for epigenomics and except for mutational level. And then the proteomics, yes, they do influence, but at the level of metabolomics, they influence a lot. So that is the reason why you and me sitting at different parts of India might be having a huge level of differences as far as metabolites are concerned, right? So what is metabolites and what is metabolomics? I have told you. Now, what exactly and why exactly it is essential, let me explain it to you. When we talk about a normal human being, nobody can know that what is a normal human being. Nobody can standardize a normal human being, right? Nobody can say this is a perfect human being. But in general, if I look into the details of the reported data, the endogenous metabolite that is present in a normal standard human being is the maximum, right? Compared to the rest, that means what are the rest? The metabolites in the form of drugs, the metabolites in the form of food additives, the metabolites in the form of drug metabolites, and the toxins, which is the least amount. So this is, if I consider that this is one of the best human beings metabolic scenario or profile, if I compare it with a diseased person, do you think there will be a difference or not? I think so. Supposedly, I'm taking the tissue of liver cell of myself. I'm a non-smoker, and I take the tissue of a smoker, liver tissue, and I go for the metabolic pathways and go into the details about what exactly the metabolites present within the cells. I think that there will be a huge changes at the level of toxins, at the level of food additives, at the level of drugs that the person is consuming. So at this particular level, when we are comparing the metabolites at a normal person and with a diseased or any other person, if we are comparing, we get a scenario that what exactly is playing a role in the changes of the organism, right? So what exactly are the factors which are influencing the metabolome? There are thousands of factors. So there can be genetics, there can be age, environment, as I have already suggested, gender, diet, lifestyle, hormones, drug treatments, everything can change your metabolome. So basically, when the doctor suggests that you have this and this disease, it's not the disease that is happening at the level of gene or that is happening at the level of your protein. It is quite possible that it is happening because of the external factors which you are facing every single day and which is influencing the metabolites in your body, right? So why is this so important? As I said, every single molecule is a small molecule, be it the drugs, be it the components of the drugs or the derivatives of the drugs, be it the small molecules in the clinical assays, be it the disorders. So everything can be understood if and only if you are going for the metabolomic study. And what is a metabolite? So basically, any particular organic molecule which is detectable in the body with a molecular weight which is less than 1500 Dalton or 1.5 kilodalton. So that is very, very small. So it can be anything. Can you see this particular data? It can be anything, starting from your peptides, oligonucleotides, sugars, nucleosides, organic acids, ketones, alkaloids, anything, drugs, drug metabolites, everything is a metabolite. And if there is an influence in that, we can easily detect by its study, right? So it is essential that metabolomics studies should be done. And when we are linking all three of them, genomics, proteomics, metabolomics, we actually get the detailed systems biology. We actually get the final answer. We actually receive that why the person or why the plant or why the animal that we are working with looks like this or feels like this or having the phenotype the way it is, right? So what are the applications? Huge number of applications. You may see that they are very, very essential at drugs and the clinical level. So it's very, very essential to go for the metabolic melomics study. So what is the workflow of the same? You can take a bacteria, an animal tissue, a plant tissue, and you go for the extraction of the same. That means you are taking out the basic extract or the biofluid of the particular organism. And then you can either go for the NMR study or you can go for the GC or the HPLC or the particular detailed study, and the final version that you received as the form of ppm or the data or the amount of metabolites that is present, that is again checked with the controlled data. Nowadays we have a lot of metabolites, we already know the RF values of the same. So when we check with them, we get to know that what this tissue is containing. So with that, we understand that how the particular organism has been influenced by the metabolic pathways, right? So this is the basic ideology, that you have to take up the metabolomic sample, and it can be any kind of sample, as I said, and you go for the separation technique. The separation technique, as I said, can be any kind of chromatography, it can be any kind of electrophoresis, where you can actually detect the value of the particular metabolite in form of ppm, right? And once you get that, it is very essential to match it with the control so that you understand that which metabolite has been present. As I said, as far as chemical diversity is concerned, I told you in one of my slides, this is the maximum that you may observe. Why? Because genomics is comprised of only 4 bases. Proteomics, only 20 amino acids can show you the results. But in case of metabolomics, it is more than thousands of chemicals that are involved or creating the metabolites that is present in your cell. And that is why metabolomics is so difficult, right? So we have series of different kinds of tests, and every test has its own level of sensitivity, what they can measure, how many metabolites they can measure, and depending upon that, we select the particular analytical technique for measuring the amount of metabolite that is present. So there are 2 routes to metabolomics basically. One is the profiling method, which is also known as chemometric, and the second is the quantitative method, that is the direct targeted method in which we directly quantify the amount of metabolite that is present in our body. So what is the profiling method? Well, we take the sample, we collect the data of the same, we prep the sample, and we deduce the sample in such a way that we get actually that which particular metabolite is present. And how they are identified, we go for the metabolite identification by matching it with the control, as I said. But by this, we only get to know that whether a metabolite is present or not, right? So we actually get to know that what metabolite is present. But when we try to quantify the same, we try to target the particular metabolite with the biological interpretation, with the biological pathways which is involving that metabolite. And when we identify and we quantify the same, it is essential for us to identify a disease cell and a non-disease cell, right? So from the spectra that we receive, we actually match it to the list, as I said, and there are series of retention factors. We have already taken time into creating these retention factors, and we have the details that which metabolite is present. So we just check the same and we identify the kind of metabolite present. And then we put them into the computational analysis to discuss the fact that which metabolic pathways was involved in creation of the same. And finally, we'll list the same to the models and biomarkers to actually suggest the fact that we started with the genome, but quite possibly that the metabolite that is present within the cell might not be responsible or might not be showing the same answer that the genome was showing. So it is very essential that we list the same metabolites, we add the same into the list of the final pathways and of the final markers that we have received, right? So this is the end of the same. I think I have, yeah, I have completed the same. And thank you so much. I appreciate your patience so much. Uh, just a second, I will stop my screen share and then we'll move ahead. Okay. Okay, fine.
Very nicely presented, Dr. Nupur.
I'm sure there must be a series of questions,
No, no,
but
actually, we
just
had closed down the window because there was some disturbance going on.
okay.
Okay, one thing before ma'am leads to the question answer round. Please, it is a request to all of you that please don't, disturb the session while it is on. You try to understand. See, one thing is that this summer training, the medium for imparting knowledge is English and will be English. If you have any problem anywhere, okay, that particular thing can be taught in Hindi, but otherwise the presentations will be completely in English. Okay, and this have we have been telling time and again, our university follows that and we are going to follow that only. So instead of wasting time in unnecessary communication, please spend some time in understanding what is being imparted. And still, if you cannot, then you are most welcome to leave and go. Okay, but it's not that we don't want to impart things, but we are trying our level best. But just to create a nuisance somewhere, please don't do that. It is a humble request to everybody. That is the reason
Hmm.
for which we had to shut down this chat box. Okay, because those who are actually trying to understand, we don't want disturbance to happen there, and it breaks the momentum of the speaker that time. Okay, so this was one thing. And secondly, regarding the test, we had told that those corrections, though no, those questions which had issues, they have been moderated, and the marks have been changed accordingly for all of you. Okay, so these were two things we wanted to share first. Okay, Dr. Nupur, as far as I can see, the first question, it comes from Harshita. Why only Arabidopsis thaliana? Why not any other plant?
The one of the examples that I gave
I think in the
back.
beginning you mentioned about the
No,
genomic
you can
study
always
happened.
take any of them, Harshita. It's not necessary. See, this is a model organism anybody will give an example of. It's not necessary that you can stick to Arabidopsis thaliana. Any organism you want to go for the detailed study, I just picked up one example. That's it. So there was no such scenario that you have to stick to Arabidopsis thaliana. It can be any of the organism, right?
Uh,
Yeah, Govardhan is saying that Arabidopsis is a Drosophila of botany, but again, I suggest that it was one of the examples. It has nothing to do with that why only Arabidopsis. It can be any of the plants. It can be even you. So at that level, at organism level, you can pick up any organism, right?
yes, and if you know the concept of model organisms, things began because that was the first plant genome which was sequenced.
Yeah.
So definitely you need sequence information to play with the data. So if one thing was available, then people started doing other things on that plant only. And if you do research in molecular biology, you will find a lot of work is done only on that plant initially. Then it was taken into different plants, as ma'am said. Okay,
Okay. The questions have started coming, I think.
that is why it will be a bit of a gap. I am just trying to jot them down also. Okay, Barkat asked, ma'am, why PCR is not preferred beside cloning, please. Don't
Okay,
understand, please explain
there
once
are
again.
certain things in which I said that I will explain. I'm so sorry I skipped out. See, there are two reasons behind that. At times, when we go for PCR, you require a prior knowledge of the same. That means you need to design the primers. Primers कहाँ से बनेंगे? उनके उसी जीन के flanking region से, right? So if you do not have a prior knowledge of the same, like you, for whole genome sequence, आपको कुछ नहीं पता, so you cannot go for PCR. Secondly, when you are going for a long stretch of DNA, you go for better methods like cloning. So you get those bags and yaks where you can actually insert a long stretch of DNA instead of going for small kilo based pills, right? And this is the reason why it's very, very essential that during the process of whole genome sequencing, people stuck to the fact that cloning should be preferred rather than PCR, right? Okay, I read what exactly caused the problem. So, Puneet, if you are there, yes, I can always speak in Hindi. Secondly, what is ATGC? I don't think this is the platform where we can teach you that what is ATGC. That won't be justified for the other students. Those people who have taken out time and actually planned to attend the summer training, thinking that it will help them out for the future, I think that will be very non-justifiable from our part to stick to the basics. You knew that this is DBT summer training, that is Department of Biotechnology. So we have kept it in a modern platform with modern techniques. We thought that the students who will be attending it will be having a prior knowledge of the same. I don't know whether we have justified that or not, but we seriously have taken a lot of effort. There is a big team which is working behind the screens. And I really take this opportunity to thank each and every member of that team who wakes up till late in the night to actually create this DBT summer training as a success. So please, if you do not feel satisfied with the sessions, you are most welcome to inform us, but not this way. This is not just justifiable. So I hope I have made myself clear. Puneet, last time also I have informed you that you should not disturb the speaker at all. If you find any issue, directly talk to us. Do not create any problem for any other participants. Okay?
And beta, you are yourself a student of BSc Biotechnology. So I wonder
Yeah,
if you
that
don't
is
understand
what is surprising for
ATGC, then
me.
how did you enroll into the course? Anyhow, that's not our concern, but the only thing is please don't create disturbance here. And especially when a speaker from outside is also coming, these things they are not acceptable at some platforms. You have to please understand that much. And this thing is for everybody. We want to impart maximum knowledge and share whatever we know with all of you, but at least if you let us know, allow us to do that in a proper way, it will be appreciated. Okay, Nupur, I
Yeah.
think we can go on with the questions.
Yeah,
So
I was actually scrolling down and that form
no, no,
captured
don't.
my
So
idea.
Barkat asked again, ma'am, how are genes recognized within the genomic sequences?
Why cannot I find? From where are you studying here?
I have copy pasted from the chat box only.
Okay. Actually, I'm
That's
searching
exactly.
the chat box. Okay,
That's
fine. What was the question? Can you please repeat? I'm so sorry.
okay. Barkat asked that,
Mhm.
ma'am, can you please tell how are genes recognized within the genomic sequences? Briefly outline, please.
Okay, see, this is the process of annotation where we actually get to know that which gene is present, what actually is the stretch of the same, from where it is starting, where it is ending. That is the complete process of annotation. So if you have the whole genome, you have got it sequenced, or you have got a lot of contegs and a lot of overlaps, you have received that, and then you have assembled that and you have got the final stretch. But then the question comes that how do you decide that what is the gene? So if you have seen in one of my images, I have shown that how the gene actually looks like. It has certain portions in the beginning, at the certain portions at the end. which are actually the recognizable and the conserved sequences, which will stay true for any of the genes, right? So these are the sequences where we try to find that probably if the ORF is present, probably post that a complete stretch of gene is present. If 5' UTR is present, that means that post that a complete sequence is present. So that is how you identify or you try to understand that the complete genome is having this gene, and then it is ending over here, and then again it is having a stretch of intron and then again a gene. So that is actually the basic thought, but then when you will learn in bioinformatics, I think it will be easier for you.
Yes. Okay, so the next question, just one second. Ayush is asking, can we say metabolite are responsible for the change of pattern of cell autogrowth to corrupted version of autogrowth, which is not suitable and cause cancer?
um,
As far as I understand, yes,
can
Ayush.
you can you please repeat?
I'm repeating. He is
Yeah, yeah, yeah.
asking that metabolites are responsible for the change pattern of cell autogrowth
Okay, okay, okay.
to the corrupted version. Corrupted, I think mutated. He
Okay,
means
okay, yeah, yeah, yeah.
mutated version of autogrowth,
It is
which is
quite
not
possible.
suitable and cause cancer.
Yeah, it is quite possible. At metabolite level, you can always look out for the changes. See, whenever you have a cancerous tissue, you will see that the changes are, see, the final version change at the metabolic pathways can be visualized at every level. So it's not necessary that at only the genomic level you can identify the cancerous tissue. It can be possible, and it is absolutely possible at the metabolic level, because the amount of metabolites being produced by a certain tissue, that will increase automatically, right? So studying the metabolites will be very, very essential to understand whether a cancer has been caused or not, right?
Okay, and I request the host to please allow the participants to unmute themselves if they have to ask any question or if it's not clear. Okay, and Ayush, I will also like to add one thing in this, that yes, as ma'am is right and you are also right, that there is change of metabolites which can cause, because any mutation that will result in change of metabolites either being produced or not being produced, and thereby the division pattern of the cell also gets hampered. But another thing is that there is also a field of epigenomics. So
Hmm.
whatever change is there which is not due to the change or alteration in the DNA sequences, some changes like that are also encountered in nature, so that comes under the word of epigenomics. So this is a mixture of both things which happens. Okay, now Sumit is asking, can metabolites help? I hope Ayush your answer is there received. It's clear. You can unmute
Yes,
and
ma'am.
okay, beta.
Mm,
Okay, Nupur, we go forward. Now, Sumit is asking, can metabolomics help to develop early detection systems to improve healthcare?
Yes, yes, of course. They are being utilized actually to, you know, it's not feasible for every lab to go at the sequencing level. It's not feasible for every lab to go for molecular level. So what they do is that normally when you go and give your blood sample, you see that they have done mass spectroanalysis or proper flow cytometry or the simple techniques. So basically, what they are looking out for, they are looking out for the normal metabolites that are being produced, to look out for the fact that what disease is actually being caused. So when you get the final analysis report at the end of the day, you see that your something has been raised and something is less and something is more, and you decide the fact reading from the internet that probably मुझे ये हुआ है, या फिर probably internet is suggesting until and unless you visit your doctor. Basically, metabolomics, when until and unless you go for the proper analysis, they actually help in the early detection, and this is actually helping the healthcare system so well, because it's not feasible to sequence whole genome for every single humankind. It's not feasible, right?
Yes, and the field of bioinformatics actually began to treat the healthcare system only.
Hmm.
Okay, and this
Yes.
application of genome sequencing to plants, this was actually an application later on. It was applied to human beings to treat the cystic fibrosis disease and everything. So basically, this caters with the healthcare only. Okay, next, Nupur. I think Urvashi has written the question incompletely also. Probably she can complete it. But I'll just read what she has written. If we take two samples of same plant, one from a normal place and one from a place having radioactive or any external factor influencing DNA, will there be an? And then it is incomplete.
You can always complete it, Urvashi. You can unmute yourself and let me know.
Is Urvashi
Urvashi?
there? Or probably we can take it up when she joins.
Okay. See, let me just hit the high. Let me see. If at all you are asking, will there be a change in the metabolite? Yes, there can be changes at every level, except for probably the genomic level. It won't be so easily deciphered. But then again, as I said, ma'am also suggested that at epigenomics level, you might see because of the environmental factors. But then as you move ahead at the transcriptomic level, at the proteomic level, at the metabolomic level, at every level, you will see that an external or the environmental factor is responsible for creating differences in the basic phenotype, right? So if you are talking about the plant, if you are talking about a sample of a plant, even that will vary because you are talking about the external factor, which is the radioactive substance or whatever you are thinking about, the stimuli that has actually created the difference. So this is quite feasible that there will be a change, but then I repeat myself, it not necessary can be hereditary or going to the next generation. It's not necessary. So this might stay to that plant of that generation only. So it depends. I don't know what your question was, but I think I have justified it. Otherwise, Urvashi, you can always ask me again.
Yes, I also think she would be asking this. It seems justifiable what you took up. And then Barkat is asking, ma'am, is there any technique to study about the post-transcriptional modification?
What do you exactly mean, Barkit? Means in the sense that when you talk about transcriptomics or when you are talking about proteomics, see all the techniques, they will tell you that there is a change. But where exactly the change is happening, that you have to go back and study. In the sense that if supposedly you are looking into the fact that there is a change at the transcriptional level or at the protein level, if you are lucky enough to do the data at all the levels and you see that at genomic level it has not got changed, at the transcriptomic level it has not got changed, but at protein level you see the difference, then of course you can, I again repeat, you can. It is a possibility that it is a post transcriptional modification, but having an assurance of the same, there is no such technique by which you can actually identify the post transcriptional modification, right? So you can justify the fact that probably the phenotype that you are observing, or probably the final metabolite that you are observing, the changes in the same is quite possible at this level. But again, as I said, quite possible, right? So you cannot totally say that this technique is responsible for this particular modification. You cannot say that in total. Am I right, Divya? Is there anything
Yes,
that I'm
yes,
missing out?
yes. No, no. And anything actually the later part of proteomics or which checks the functionality of the gene. There are techniques to, there are kits available to work on gene silencing aspect and everything. So all that is post-transcriptional only, which happens, included with what ma'am just said. So I think, uh, are you satisfied, Barkat? Whatever you were asking.
See, when you write a publication, you must have seen that in research papers also, when you are finally concluding a particular statement, you always write that it is a possibility or probably, until and unless you are 100% sure, you cannot give the statement that this is the reason behind this change, right? So there are thousands of analyses by which the probability ratio increases, of course. But again, saying it 100% that this is the post-transcriptional modification because of which this thing has happened, this is quite rarely found, right?
Okay, Barkat is okay with the answer. That is what he's written. And then the question is from Anisha. Ma'am, how overlapping small segments of DNA are created as the restriction enzyme is cut at specific sites?
Okay. See, this I think you might have studied before also. Whenever you have a restriction site, it is quite possible that within that restriction site, another restriction enzyme can work upon. It is quite possible that when we are looking into a vector system, there are a series of sites where different kinds of restriction enzymes can work. So that whenever we are using a particular restriction enzyme, we get a fragment of a particular size. But when we are using a different restriction enzyme, we are getting a different fragment. We are looking out for overlapping fragments, right? So it is quite possible that when we are using different kinds of restriction enzyme, they are cutting at different sites and they are giving different kinds of fragments. But we are essentially looking out for the overlapping fragments where there will be certain similarity at the ends of the fragments, right? And this is possible when there is chances that two restriction enzymes are cutting within a big chunk of a DNA. So that one fragment will be creating one particular, one RE will be creating one particular fragment, the second will be cutting the same site but at a different place, then a second fragment is created and then the overlaps occur, right?
And yes, Nam very rightly said. So whenever we do work like that, genome walking or chromosome jumping or anything, so we whenever we create libraries, be it DNA, so we use combination of restriction enzymes. It's not only one which we use. So a combination, and thereafter we generate different libraries with different enzymes. And then go on with the work. And I think probably because this is the first session on omics and maybe all students are from different levels, so probably by the end of the week, when you get exposure to these topics in detail, some of them, so that will be more clear. Okay, so the next question is from um,
I, I,
Urvashi.
I think yeah, Urvashi has
Ah,
completed her question. Uh,
um,
she is
First one, how
right.
do we come to know which one is unmuted one if the results got
Okay,
mixed?
she's saying that uh, so while sequencing, how do we come to know which one is the unmuted one if their results got mixed and the plant is unknown at biology? Okay, Urvashi, do me a favor, dear. Can you please unmute yourself? Please, please.
Is Urvashi there?
Urvashi,
Yes,
please. Yeah, please explain me the question, dear.
ma'am, I'm asking if we are taking sample of a new species and one from the normal place and the same plant species from the um, mm, so-called like places like Japan where there are radioactive effects on that plant species
Mm-hmm.
or other factors affecting the plant DNA. So,
Mm-hmm.
and we do the sequencing of the DNA in the laboratories, and
Mm-hmm.
the result got mixed up, and we have to know the which one is
Mixed
the
up
correct
means what?
sequence.
Means the samples have got mixed up.
Yeah, and I mean the labeling got from which place got it was taken, and we don't know which plant it was, and it's a new species discovered.
New species. Okay, okay. Please come
Yeah,
again.
ma'am.
You said that there is a mixing up of the sample, and you
Yeah,
are
ma'am.
confused of the fact that whether a new species has been found or not. Am I right? Is this what you are asking?
N- No, ma'am. I'm asking that the species is new, so we don't know about anything about the sequence before the sequencing is done. So we are doing it for the first time,
Mm-hmm.
and
See,
we got one muted one and one normal sequence. So how are we supposed to know which one is muted and which one is
Urvashi,
original?
this I am answering totally based upon the fact. First thing, nothing like that happens. Second thing, if at all, which is 0.001% chance in a molecular lab, because the people who work at the sequencing level, they are so limited in numbers because of this reason. Because we do not want any kind of mixture. We do not want any kind of unlabeling of the things. This is considered to be the biggest hazard in case of molecular labs, right? So even if chalo ho gaya, मान लिया कि हो गया ऐसा. So thinking the fact that if something like that happens, what I will do is that first thing, whenever I take a plant genome at bioinfo level, I have always observed, and this has been reported as well, that there is a 99 point some percent identity between the plants, be it between the species, be it between the genera, because they are plants, right? So now coming to the fact, since you are talking about nucleotide level, I'm not going to the level of proteomics. I'm talking only about the sequencing level, right? So if at the nucleotide level, I do a blast, or I try to check out the genome of both the data, then there is quite a possibility that I will get an answer, or I will get a hit with 100% possibility of matching to a standardized genome sequence that has already been reported, right? So with that, I will identify that one of the plant is quite possibly the standardized plant which has been grown in the normal conditions. If at all I find even 0.5% variation or gaps or unhits or missing areas in the second sample, it will be a probability that it is the second sample which has been developed in the areas which you said that radioactive it is or something like that. But as I said, at genomic level, it is very, very difficult to identify the normal plant and the plant which has faced the external stimuli, right? So if you have mixed it, first thing, you won't be allowed to go for the next level. I'm telling you
Yes.
very frankly. So, secondly, even if you have missed and you have not reported to your guide and you want to find out that where exactly I'm going wrong, this is the way you can try to find out. Third thing, I would suggest go for the higher level of analysis. Only then you will get the answer. You will never receive the answer at the sequencing level. Never ever. So until
Yes.
and unless you know the differences at the protein level, at the metabolite level, you cannot find out that which is the normal plant and which has faced the external stimuli. Okay, Urvashi, am I right, Divya?
Yes, yes. And before we, we publish, it's not only with one technique we have to find this out. We have to prove it with many techniques. So if we are checking upon mutation, your site-directed mutagenesis and everything, we'll have to do and conduct and compare. Then only we can say this is just with the first thing of sequencing. We cannot say. And as ma'am said, there is some concept of orthologous and homologous and paralogous. So homology is something in evolution. So there is a lot of similarity between the genomes of organisms, even if they are from different kingdoms altogether. Okay, so if you want to see the pattern, yes, in at some way you can, you can get a hint if there is a mix up, but still leading on with the wrong hint at the first step, it is a very what
It's a
it's
big
a suicidal
disaster.
step.
Yeah,
Yes,
yeah, it's a
for
big
the
disaster.
research. And PhD is not a matter of just 1 year. That's why it takes time, because any research you have to do, you have to prove with as much evidence as you can. So, as ma'am said, you not only at the level of bioinformatics you'll prove that, you'll also prove it at the level of your, what do you say, transgenic study. You try to create a mutation yourself and see what is the influence, and then only you correlate and support the data together, and that is how publish your data. Okay, Urvashi?
Yes, ma'am. Ma'am, one more question.
Yes,
Ma'am,
Urvashi.
like COVID-19,
Hmm.
some say it's reverse engineered and some say it's the original one. So how are you supposed to find out at the genetic level that either it's reverse engineered or it's the original one and not the muted one?
Okay, I read an article related to this, in which a group of scientists, they are working on, that particular sequence which is there in the coronavirus, which is mutating, and they have found elements which naturally is not present in viruses. That is why they are saying it is genetically engineered. Yes, Nupur?
Okay, ma'am.
Am I right?
Yeah,
So
yeah,
that
yeah,
is
yeah.
that is
Actually, I
done
was
with the
reading the
help of,
questions. So sorry.
okay, okay, no, no, that is done with the help of bioinformatics only, and a lot of molecular experiments are laid out, but they have found some regions of regulatory elements which are naturally not present in viruses. So if you you use the tool of BLAST in bioinformatics, so you get homology. If whatever sequence you put of the DNA, you can get its homology related to whatever sequences have actually been worked upon. So whatever viruses have been worked upon, they analyzed data with that, and they found that those regulatory regions are not naturally present. So they are hinting that it was created inside a lab. That is the reason. This is
Okay,
as
ma'am.
far
Thank
as
you,
I
ma'am.
know right now. Rest everybody is working on coronavirus. So we get to know
Yes,
things.
ma'am. Thank you, ma'am.
Okay. And Bhupen is asking Nupur, ma'am, could you please show and explain the slide containing several instruments for determination of metabolites like NMR and GCMS?
Yeah, Bhupen, I actually thought of doing that, but then you have one complete week of metabolomics in which you will
Yes.
be explained about the
Taught
same.
all this.
So you will be in a detailed form, you will be taught about the same. I think it's better you attend those sessions. It will be difficult for me to explain in such a short time. So kindly wait for one more week. You will be getting all your answers regarding the same.
And then Kanupriya is asking, ma'am,
Ma'am,
what is the solvent used for metabolomic study? So there are different combinations of solvents, and as ma'am said, you will be given exposure to this. So I hope Bhupen and Kanupriya, you will get your answers in the coming week. Is it okay?
okay.
Okay, Suraj is asking, do these modern fields of study nullify our existing genetic developmental theories, gene centric pathways of expression, or do they coexist with them
Okay. Or they supplement them.
or supplement them?
Gene-centric pathway of expression. See, I haven't changed any of these theories. Whatever you have studied today or whatever I have tried to explain today goes parallelly to your molecular biology concept that you have studied in your NCRT. Nothing extra has been told to you. All I am trying to explain to you is that at the level of omics, what exactly your exposure is like, how they are going to change the scenario in today's world. See, the pathways of expression stays the same. In your NCRT as well, you have studied that how the RNAs, how the proteins are, the expression levels of the genes that you have studied at DNA level. So why will I change that? Means I really cannot understand. I'm not supplementing it. I'm not coexisting them. I'm supporting it all together, saying and suggesting that what all possibilities are there and where exactly we are lacking and where we should move towards. That's all I did. I think I have justified it. I hope so. I have justified that.
Okay, and
There is
next is, what is the cRNA? Murgeshwari is asking.
see RNAs. The way I have no clue. Do you have any idea?
No.
Can anyone tell me exactly what
Murgeshwari,
is
can you
cRNA?
please
Yeah,
clarify on that? Okay,
It must be cDNA, I think.
I think, ah,
if it is cDNA, then I can
it's
answer. If it is
complement.
cRNA, I really cannot answer because I have no clue. Yeah, just a second.
Ma'am,
Yeah, I'm so sorry, I had to take a call. You wanted to say something, dear?
No, no, I think that is what if. Yes, yes, it's cDNA only. She's
Okay,
writing, and
okay. So when we actually go for reverse transcription, I hope you know what reverse transcription is. When we are replicating a DNA, that means we are producing DNA out of DNA. When we are going for transcription, we are creating RNA from the DNA, right? When we are going for reverse transcription means we are going back in the central dogma. That is, we are going to create a DNA out of the RNA that has been created. Now, if I am doing that, or if somebody is doing that, then what is the benefit of the same? The benefit is that we are actually getting a modified version of the DNA. It is not the normal genome that we were talking about. We have created the DNA out of the mRNA. So that means that all the changes has happened. Splicing has been done, all the modifications has been done, tailing has been done, and everything has been done. And now I'm creating a DNA out of it. Now, why do we go for that? See, this kind of enzymes were essentially found in the viruses, and it actually explored a new era in which we could create a newer version of DNA out of the RNA. And since it was giving us the better answer from the mRNA, why? Because it's not easy to work with RNA at lab level because it gets degraded so
yes,
fast. Even your fingertips has RNAs, right? So in that scenario, we require a stable form of nucleic acids, and what better way than to use cDNA, right? So this is the complementary DNA which has been formed out of the RNA molecule via the process of reverse transcription,
yes. As
right?
ma'am rightly said, RNA is very labile. You can't work with RNA. DNA is very stable. Even at around 38 or 40 degrees, if you keep the DNA just on your table, it will nothing will happen to it. So thankfully we know the process of reverse transcription, and thankfully we have the reverse transcriptase enzyme also isolated. So we use that for creating cDNA. Okay, and then Ayushi is asking, ma'am, from where I can get? Oh, okay. For the related to the test, I think you can put down the email on the email ID which has been provided to you, right? So that is if that is your query, because we are not looking at that part. So right now I don't have the idea. So please put your query there. And then Kanika is asking, what is the best assay for identifying an oncogene or any oncogenic domain? Does transcriptome data contain introns?
Okay, I think um, this is Kanika.
Yeah, Kanika.
Okay. So, oncogene, basically, when you are having a person who has been diagnosed with unfortunate cancer, so basically, we try to go for further analysis with the tissue that we are having in hand. So, if at all we are receiving the tissue from the person, see at the level of blood test or MRIs and NMRs, we get to understand the fact that a person is carrying cancer. But if at all we go for the process of biopsy, that means we are extracting the cells of the same and we are going for the further details. If at all we are capable enough into going into the details of finding the gene at which level it has got mutated or at which level it has got affected, then we of course can use any of these analysis depending upon the fact that you can start even with sequencing, the basic sequencing, because nowadays most of the cancers have been identified, sequenced, and their gene has been located. So it's very easy that you can always go for sequencing and you can always find out a domain that which is overexpressing or less expressing. You can always go for the transcriptomic analysis, you can always go for the proteomic analysis. At any level you can go and check. But as I said, it's not necessary that you can afford that. It's not necessary the patient is willing for the same, right? So if I talk about at the moral levels, I don't know how much that is feasible or how much they allow. But if at all a lab is capable enough, they can go for any of the levels. And since you are talking at the gene level, you can identify at genomic level as well. So it's very easy. Once you sequence it, it's very simple. You use the bioinformatical tools and you can blast it and you can find that whether a identical match has been found or not. or you can always go for the higher level for transcriptomes, or you can always go for the higher level for of proteomes. So you can always identify whether a gene has been expressed or not. If it is being expressed, how much it has been expressed. So by that you can also understand the fact that at which stage the cancer is. I think, Divya, is there any other essay you can think of?
No,
Because I
basically these days, using bioinformatics tools only in silico analysis, we'll try to differentiate between the oncogenic domains or
right.
oncogene. So, and that, as ma'am has said, thankfully a lot of work is going on. So, a lot of data is already there. And somewhere the same concept of homology comes up, even if some data is not there, some gene is very new. You know, new doesn't mean that it will be a combination of somehow ATGC only, right? So there will be some homology data which would be available, because many times even your some regions in the retroviruses, because they undergo mutation very fast, and those sometimes if we use the bioinformatics tool, we find regions of homology associated with retroviruses. So somehow that those who are working on it, they relate it to some sort of mutation going on. So mutation is also one reason why cancer happens. Okay, one of that. So I think majorly at this point where molecular biology is, people would employ the use of bioinformatics only to aid with it, and definitely checking out the functionality of genes. That is how I would go about it.
Yes, you associate the data with the phenotype and then you easily get the response. So, yeah. So there is one more question from you, that is, does transcriptome data contains introns? I think I answered that in my slide itself. See, transcriptome is what? This is the RNA level. RNA means what? That all the splicing has been done. Everything has been, removed out of its way so that you are left with only the open reading frame or only
Yes.
the that area. So no, there is no chance that in transcriptome data you get
And splicing, the word, if you see, it does not mean removal of introns, it means
right.
joining of exons.
Exactly.
So the definition itself says when it is joining exon, then why should it have intron data? Okay, so I think now, Nupur, I will take up the questions together.
Yeah, yeah.
Same person has, many questions that is happening somewhere. Like Barkat is asking, ma'am, briefly outline how mutagenesis screen is carried out and how genome difference correlates to species.
Okay. I can I okay, I see. Okay. I found the question. Fine. Why can't genetic test? Okay. This is
No,
Barkat, right?
yes, yes, at
Okay.
334.
Right. I have received it. Okay. How mutagenesis screen? Okay. So, see, whenever you are going, you again, as I said, mutational analysis is what? Whenever you are planning to go for sequencing of not only the complete genome, if you can afford, of course. So, if you want to go for mutational screening of the same, let me give you a very simple example. At times, what we do is that we go for reverse genetics of a particular organism. Supposedly, we have a particular plant. Suppose a rice variety is there, which is very wonderful, but we cannot afford the sequencing, the whole genome sequencing of the same. My lab is not so proficient of the same. So, what we can do is that we can always create mutants out of the same rice variety by chemical treatments and all the detailings, and it takes many years of actually growing and regrowing of the particular population till the moment we find that the mutation has stopped, in the sense that it is not segregating anymore, it has stopped and found its place. Then it's very easy to actually find the phenotype, and we start locating the fact that whether the phenotype is matching with that particular mutation or not, because my rice variety is having the best yield and the best condition that I started with, but suddenly the mutant that I have created, that is having means probably very strong roots and better yield. So I think of the fact that probably the mutation is the reason behind the same. Now it is easy for me to create or to go for the sequencing of only those genes which are related to those mutations, right? So when I do that, I find that yes, after of course the computational analysis, I find that yes, there are single nucleotide polymorphism, or there are probably long stretches of genes that has jumped from its place, or probably there are mutations of a big chunk, and I associate the same with the phenotype that I have found on my field level. So this is what this is the normal screening of mutations that you do at the breeding level, right? And it's easier, but it is tedious in the sense that it takes many years to finally stop the mutation from jumping from one place to another, or to stop segregating the mutation. So that's why we need many populations to finally decide the fact. Now, this is the normal process by which you go for the reverse genetics and you associate the same. You can always go for other processes where you can completely mutate every single gene of the same, which is quite tedious, but then again, it is not very foolproof also, because it's not feasible that you can study each gene. But if your lab is very, very rich, go for a whole genome sequencing. You will get the whole genome after the mutation. Go for the blast. You will see that where exactly the mutations has taken place. And then you check out with the phenotype that has changed in the particular organism. So basically, when you screen for mutations, it is very essential that you get the fundings and you get all the sequencing done at a particular level. And of course, it's very important that mutations, whatever you are receiving, whether it is hereditary or not, whether the mutation is showing any phenotypic changes or not, right? So all these factors are essential for mutagenesis screening. Barkit, Barkit, is it okay? Did you understand what I have tried to explain?
Okay, ma'am. Uh,
Okay.
that person, then you can always, as ma'am said, this is one step. Second step is site directed mutagenesis. That's also a technique which we do, in which we target some portion and check out whether it has got mutated or not at the end with the phenotypic level. Thirdly, when we create transgenics, there are concept of reporter genes and marker genes, if you know. So they help you in asserting if mutagenesis is occurring, okay, and or mutation is occurring. And then something known as alpha complementation test or blue white screening test, so probably you will get exposure to all that also. So for even solving one problem, there are many methods in which you can use together to justify it, and justification is done with as many proofs as you can. And then how genome difference correlates to species. Okay,
Yeah, Divya,
exactly.
Divya, so sorry. You are talking to me?
No, no, yes, yes, I'm talking to you.
So
Sorry.
sorry, so sorry. I was just reading the questions. Yeah,
No,
yeah, please,
no.
please.
Now, okay, I really don't know. Barkat is a he or a she. I'm sorry for that. So that is why I'm again and again saying Barkat only. So, how genome difference correlates to species, and this topic is actually not related to session, but I think I want to know if I want to check an anti-cancer value of any phytochemical extract, how can I? What do you mean by anti-cancer value, Vasit?
Yeah, who has asked this question?
Barkat only has asked at
Okay, okay,
3:48.
okay.
I've grouped the questions together so that we deal with one person at a
Okay.
time.
Hello, ma'am.
Yes,
Ma'am,
Barkat.
hello.
हाँ हाँ
Yes,
बोलो.
beta, bolo, bolo.
Ma'am, I mean, ma'am, if we extract any phytochemical extract from, like I extract some chemicals from saffron, Crocus stylus, and I want to see they have anti-cancer value, whether they are used against the cancer. How we can use these chemicals against the cancer, and how we can determine that it has anti-cancer values.
So you test it on the model organisms. So you take for human, if you have to do anything related to human, you can't work on humans directly due to ethical laws. So on mice, on guinea pigs, on those organisms, the extracts are checked. So you have cancer cell lines also. One thing is work on model organisms. Second is you have cancer cell lines like HeLa cells.
HeLa cells, yeah.
Yes, so you take those lines, grow them under tissue culture, have proper cultures of those cells, and then with your phytochemical chemical extract, use different quantities over those cultures and see what is the effect on of the chemical. So if it is a chemical that will work on cancer, then it should probably kill the cells or stop the growth of those cells. Okay, so HeLa cells were also extracted from the patient who Henrietta Lacks was the name, and she had cancer. So the cells had been extracted from that patient, and sequencing work and everything was done. And thereafter, that cell line has been maintained. And people who work on that, they take lines which are related, uh, which are similar to that, or that line also. Okay, so then Gobardhan is asking, ma'am, why benign tumors does not spread like cancerous tumors? What is the molecular difference or molecular structural difference between these two, which makes one spread rapidly and one stops spreading?
Divya, I really have clueless about this one because I'm
Okay,
not into
I
cancer.
see. First, even I am not into cancers, and secondly, I'm also not in zoology. But still, I will try to, answer according to the knowledge whatever I have. So, any the first question is why benign tumors don't spread like cancerous tumors. Some mutations are like silent mutations which may be happening. So mutation, if it happens at those portions which are or those genes which would be responsible for multiplication, or which is involved in the mitotic division of the cell, if that is the case, then it would not spread anywhere according to my understanding. Okay, and what is the molecular difference or molecular structural difference between these two which makes one spread rapidly? And okay, I am not very sure about the molecular difference or structural differences because I've not worked on these lines as such. But according to me, if the mutation happens at those portions, see, that is why even this vaccination step for coronavirus, people are saying that it may work on different strains, it may not work on different strains. Now how they check if it works or it doesn't work? They check by using different concentrations or different vaccines on those lines, right? So similarly, even for your okay, and if a vaccine is being produced at one region or in a manner in which it does not affect region which is being mutated, then it should work on all the strains. So similarly, if a gene which is involved in the mitotic division of the cell, that is getting mutated also, that would stop the auto-regulation of the whole pathway, and then your excessive divisions happen or they don't happen. Okay, the person is also writing something else probably.
You can always unmute and answer us, please,
Very different.
whatever you are feeling like, because that will be a kind of discussion rather than one-way answer.
Yes, because we are very clear with this. We are not into those. We don't want
See,
to
we are
impart.
all are botanists over here. We never ventured into this field of oncogenetics, and of course, this is very interesting. That's why we even want to learn about it. So it will be very good if you can
And it
unmute yourself and
yes, and it
explain
should
it
be a
to us.
two-way interaction so that we also
Exactly.
benefit somehow.
Ma'am, I am also from botany department.
Okay,
Yes,
Ma'am,
beta.
Beta.
ma'am, I saw some slides about oncogenes and cancers, so I
Mm-hmm.
asked this question. Even
Mhm.
I saw somewhere that benign tumors are non-cancerous tumors and it's non-harmful. So it's getting like uh, half of the step, but it does not spread. That's why the question occurs here. That why it does not spread like a normal cancer cells.
So then, why
That's
has
why
it become a tumor if it is not spreading? Even if it is
ma'am,
benign, but it is a tumor, right? So what exactly has happened?
this is also my questions. Why it does not
Okay,
spread
okay,
like
okay,
a
okay. This is your question. So sorry. I thought that you are explaining it to me. Okay, 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 fine.
no, ma'am. I have no explanation of this.
Okay, okay, fine.
Okay, beta. I think we would like to read. Probably tomorrow you can remind us on that, and then probably we'll
Ma'am,
end.
can I? Ma'am,
Yeah,
ma'am,
please,
Yes,
can I
please,
say
yes,
Sarkis.
something?
Yeah, go
yes,
ahead. Go
beta.
ahead.
Ma'am, there is difference between benign tumor and malignant tumor.
Hmm.
Ma'am, in benign tumor, because it's the mutation in cells and cell like their cell size, as you all know. But ma'am, in benign cells, there is well differentiation. That means they are differentiated according to their fate. But in case of malignant, this differentiation is lacked because the cells are not attached to their filaments, cytoskeleton, because they lack cytoskeleton and they separate all over the body in case of malignant case. But in case of benign, they have some control on them. And cytoskeleton system, they are attached with cytoskeleton system, they are attached with basement membranes. And when they got detached from basement membranes, and they get separated all over the body when they get malignated. Okay, ma'am.
No, yeah,
Uh-huh.
but still I
Yeah,
also
even
want to
I'm against
search
the
on that
question.
more.
Ma'am, may I say
Probably.
something?
Yes, beta.
Yes, please.
Ma'am, actually, benign tumors are encapsulated and they have their self-defined boundaries, which malignant tumors do not have. That's why their cells are unequally distributed, so that they can spread all over the body through, you can say, the fluids or other material, and they show metastasis, which is not found in the benign tumors. That's why they are
Now the
opposed to one
question is that
origin.
if Tanuj and Govardhan and Barkit, if what I could understand that,
मैम मैं थोड़ा
हाँ Barkit दोनों,
मैम मैं थोड़ा हिंदी में बोल सकता हूँ?
हाँ हाँ
हाँ हाँ
बिल्कुल।
बिल्कुल,
मैम जो बेनाइन ट्यूमर है ना, ये इसके जो सेल्स हैं, ये सेल जब बेनाइन स्टेज में होते हैं तो ये बेसमेंट मेम्ब्रेन के साथ अटैच्ड होते हैं। बेसमेंट मेम्ब्रेन में साइटोस्केलेटन सिस्टम का एक पार्ट होता है, तो उसके साथ अटैच्ड होते हैं। लेकिन जब इनका पूरा कंट्रोल लास्ट हो जाता है तो मैलिग्नेंट स्टेज में चले जाते हैं, तो बेसमेंट मेम्ब्रेन से डिटैच्ड हो जाते हैं, और मैम फिर ये ब्लड में भी चले जाते हैं और बाकी सभी ऑर्गन्स में चले जाते हैं, और ये फिर सेपरेट हो जाते हैं क्योंकि इनका लास्ट हो कंट्रोल जो है वो लास्ट होता है, और फिर पूरी बॉडी में जो सेपरेट होते हैं और मेटास्टेसिस हो जाती है। लेकिन जब ये बेनाइन स्टेज में होते हैं तो ये अटैच्ड होते हैं बेसमेंट मेम्ब्रेन के साथ। मैम थोड़ा इसके बारे में आप भी पढ़ लीजिएगा, बेसमेंट मेम्ब्रेन के साथ अटैच्ड होते हैं, तो फिर ये कहीं भी जा नहीं सकते हैं, वहीं पर अटैच्ड होते हैं, और मतलब वहां पर जो है इनका कंट्रोल थोड़ा सा होता है। फिर जब ये लास्ट कर जाते हैं और बेसमेंट मेम्ब्रेन से चले जाते हैं तो फिर ये तमाम बॉडी में जो है फैल जाते हैं। इतना नॉलेज थोड़ा सा।
okay.
हाँ,
So but the question stays molecular difference.
yes, this I can buy the fact that encapsulation, at some point it stops the spread, or it doesn't contain the spread. But yes, there would at molecular level, I think it would have to do with mutation only, because p53 and every, there are tumor suppressor genes and everything also present. So this is a regulatory network which happens. So cancer is just not only one gene got mutated and you had cancer.
No,
So
there are several
yeah,
oncogenes.
this is
That
a whole
I agree. But then
regulatory
how the
network.
difference is occurring at the benign and the malignant level, that I am not sure of. That's why I was asking.
Right now we are not sure, but we can buy the statement, yeah, about the
But then they are
encapsulation from
answering at the
Tanuj
cyto level, no?
at the
Cytogenetics
cyto
level.
level. But at
Exactly.
molecular level, I'm sure there would be a
There
difference.
must be
There
several
would be a switch.
differences. Yeah, yeah. There must be a
Either
regulatory switch
yes,
regarding the
yes,
same.
yes. So there should be. So probably we can also search on that. You guys also can search. Everybody can search, and we can also discuss this
Yeah,
in
we
the
can have
coming
a
session.
discussion regarding the same. It's an interesting topic. See, this is something that there must be so much work that have
Still
been
going
done.
on.
Yeah, there must be, and there must be many regulatory stops and many regulations that are happening at one point or the other, which is leading to the molecular difference. So,
Hmm.
at what level that I have to go for generous amount of study? That's
So
true,
because
but I would
cancer is something that the proper cure has not still been found, and that
exactly.
is not been found because there are differences at the regulatory level. There is different networking which happens. So we, as a scientist, a person should be able to control each switch. Then only you can, control the cancer properly as such. But yes, we'll like to search on this more, and if we come across some research papers, we will share it on the Telegram group.
Yes, yes, of course.
Okay, Gobardhan, because beta
Okay,
at
ma'am.
this point even we cannot give the statement because we are not well qualified to do that right now, because we need to study for ourselves. this thing. We are also students throughout life. Okay, so now Tanya is asking, ma'am, how does metabolomic act as a primary factor for the field of personal medication development?
See, I told you before also that if your metabolites are differing from a healthy person, which is again a statement of vague idea that what a healthy person is. But then again, there must be some criteria or a standardized person's metabolite profile with which you are comparing with a diseased person's metabolomics. So once you get that, automatically it is easier to understand that at what level the pathway is differentiating, or the pathway is not taking place, or probably there is some sort of stoppage at the one of the levels. So this is easier for developing medicines by the drug developers, so that those pathways move on. I am forgetting one example. I really am clueless. There was one medicine which became very popular for actually pulling ahead the pathway, and because of that the final product that was being stopped in the diseased person that moved on. So these are completely attached to each other. So whenever there is a drug that is being designed, it can be at any level that they can help out. So whether it is metabolomics or whether they are metabolites or the particular pathway they are working on, it's easier to go for the drug designing. And personal medication development, I'm not so sure. Personal means your own, means you are going for home remedies or what?
Ma'am, by personal medication means that the concept of personal medication which is coming up instead of
What is personal
generalized
medication?
medication.
What is
Ma'am,
personal
like
medication?
if a person comes with a particular symptoms or a disease, their genome sequencing is been done or whatever at various levels, and the doctor suggests them that particular medication also, which is targeted to for that particular gene or the disease.
But then you are saying at the genomic level, not at the metabolomic level. At the metabolomic level, of course, you can go for the same. But again, as I said, something at this level, how much it is feasible, that is different for every single scenario. I don't know exactly at
So
our.
I didn't understand the meaning of personal medication here, Tanya.
He is saying that every person will be having a different kind of medicine depending upon the kind of metabolite difference
Okay.
that person is carrying. That's what she is meaning.
Okay, but for this, there has to be a creation of those medicines in that manner.
Exactly. It will require a lot of money to be spent to create your personal medication. Whenever
Yes,
a doctor
ma'am. This is
treats
a
you, yeah, whenever a doctor treats you, they treat you based on your symptoms. If you're lucky enough, they will treat you based on your omics or one of the omics. If you're lucky
But
enough, means you are having.
that is mostly at the level of something like when you have mutation like you check for Down syndrome, like all the pregnant ladies during the ultrasound, they are checked that the child they are carrying, that person shouldn't have any chromosomal abnormalities or anything. So there are particular marker tests for that. So at this level right now, I don't think that personal
There is
genomic
no
medication.
personal medication development. Means even if there is, it is. I would be very surprised. Tanya, do you have any example of the same?
Ma'am, it is, we were taught in our classes like it's an emerging field where
Mhm.
the patient is being targeted based on his or her particular symptoms only. Like,
Symptoms, yes, dear, symptoms. Symptoms, absolutely true. I do not deny that. But then when you are saying omics, I really cannot judge that, because every person's metabolomics will be so different from the other. How can you create a personal medication develop means
हाँ
just
मतलब
on.
it depends. First is, money is a very important factor in this thing, because as you were saying earlier, there was a concept that you do the pathology test, taking the blood sample of the person, and then see, analyze it, and then you come upon the disease thing. Yes, these days, yeah, even in US, people go for this really pedigree analysis as such. Not only in US, pedigree analysis can be
Here
done in
also we are going for that,
anywhere,
right, right.
but in US normally also people can go and get it done. So usually that thing we will not, as I don't know, this culture is not probably come up now, that we won't go and see how related we are to anybody.
I'm not so sure, Tanya. Quite possibly in Ayurveda or homeopathy, there is chance that depending upon your symptoms,
No,
you are creating
homeopathy,
a, yeah.
but homeopathy is also symptomatic treatment.
Right, right. That's what I said. It's symptoms, not metabolomics.
That is
As I
coming up, but in diseases which are of, grave concern, something like Parkinson's, something like Alzheimer's, which actually not a normal, disease.
right.
So genetic sequencing and everything would be done, but at that level. Because if you would realize, when you will see, if for just you get 500 or 700 base pair sequencing done, okay, of any sequence, that costs around 500 rupees. So human being has 3.4 into 10 to the power 6 base pairs of DNA. So if you want to get the entire stretch sequence, just
It
think
will
how
be very
much it will
costly.
require. Secondly, people who go for sequencing or analysis, usually they mark some regulatory regions according to which they see the differences in organisms or something, right? So yes, this is an upcoming field that people are going for it, but it is normal, not for normal fever or cold and cough. This kind of medicine will be offered. This would be offered for cases of grave genetic diseases, okay. And but personalized medicine, yes, personalized in that sense that the doctor will see it and doctor will only prescribe it accordingly. So normally for the diseases which are related to genetic disorders, so they have cytological preparations they may be making or something they would be making, then only they get to know that person. So at some point, yes, some sort of medication like that would be happening, but not for the general pattern of diseases. This is what I understand.
Okay,
Okay,
ma'am.
okay, Tanya. So then Kirti is asking uh, that can you please tell, she liked the session very much and she is saying that can you please tell names of some related books so that we can read more details from it.
I think,
I think probably they can go for genes. They have a
yeah, they can
series
go
of
for
genes.
cell and mol bio books. At
Yeah,
least
but
the basics will be clear with
yes,
them.
cell and molecular biology related books. Then there is a book known as Genes by Lewin. There is a whole series. You can go through that. Then there is Genome by T.A. Brown.
T.A. Brown, right, right.
That
Even
is a
that
very
is
good
good.
book. And Molecular Biology by Watson.
Yeah. Go
Kirti,
for
I
them.
hope we are jotting down these books
In fact, anybody who wants to go for any further studies or detailing of the same,
related to this.
please
Hmm.
refer these books. They are very helpful. They have
Very
helped us a
good
lot.
books. Yes.
Yeah.
Okay, Kirti. Okay. And then we have a few questions from Puneet. So probably Nupur,
I think, Puneet, you can unmute yourself and tell your questions yourself. I think that would be good. Puneet, are you there? Can anyone tell me, is Puneet there?
I'm checking myself.
I think he has left this
Uh,
meeting.
I also think. But the pattern of English in which he's written down the questions, it doesn't seem that he doesn't understand English.
And if
So
the
I
questions
don't.
are so
I
well formed, then I think he knows what ATGC is. I hope so. He
So it is
knows
just to
that.
create some disturbance in the
Oh
session. But
God.
seriously, the pattern in which he is writing, I can't accept that he doesn't understand English.
Oh my God. Why do they do this? Anyhow,
So
I hope we will meet Puneet again tomorrow, and I will surely take up his questions. I will be there, and I would love to entertain his thoughts. So, I
if
think,
anybody else wants to know anything which he has asked, you can say. Then we
yes,
will,
please. Yes, please.
discuss
Let us
those
know.
things. Otherwise, the person who's written down the questions, that person is only not there. So we
No,
don't
no,
want
no.
to.
I want to talk to him directly. This is the reason I won't take up his questions until and unless he comes himself. So I won't actually entertain this thing.
But
And
anybody else wants to ask related
anybody
to
else
anything,
wants to ask us anything related to the topic of today, yes, you are most welcome.
Okay, so I don't think anybody is writing.
Okay, I think that will be fine for today, Divya.
Yeah,
Thank you
then we will
so
call
much.
it a day and we stop it here. Thanks to everybody for joining.